All of the excretory tubules have an initial collecting region where primary urine is formed, then one or more region in the tubule where primary urine is further modified by either addition or removal of particular solute. Many of these tubules include an even more distal area in which the urine can be made more concentrated or more dilute than the internal body fluid. So, in insect, we have seen that the Malpighian tubules are present, and in them, in insect as well as several myriapods, they have excretory and osmoregulatory system based on Malpighian tubules and rectum. As already I have told you, the Malpighian tubule and rectum composite in which urine formation and reabsorption occurs in separated structures. Production of urine occurred by secretion in the Malpighian tubule cells. The hindgut of insect has various specialization to effectively reabsorb water and also to regulate the combined composition of urine and feces. Amphibians, they have inherited an essentially freshwater adapted kidney from their ancestors that can produce only hyposmotic urine. These animals have the ability to decrease urine formation rate and also increase urine reabsorption process from the bladder with the action of amphibian antidiuretic hormone ADH hormone. It can also regulate their osmotic balance with active salt uptake through the skin. एम्फीबियंस के अंदर स्किन के द्वारा सॉल्ट अपटेक पॉसिबल होता है जिससे कि ऑस्मोटिक बैलेंस जो है वो बना रहता है। इसके अलावा एम्फीबियंस जो कि पानी में रहते हैं, कुछ समय ज़मीन पर रहते हैं, इनके किडनीज़ इस प्रकार से हैं कि वे जो यूरिन प्रोड्यूस करते हैं, जो मूत्र उत्पन्न करते हैं, वो हाइपोऑस्मोटिक होता है। सॉल्ट की कॉन्सेंट्रेशन यूरिन में कम होती है। रेप्टाइल्स आर नॉट एबल टू प्रोड्यूस hyperosmotic urine even when they are placed in extremely dry habitat. They produce isoosmotic urine and excretion is excessive. Salt occurred through their nasal salt gland. Reptiles हाइपरऑस्मोटिक यूरिन नहीं बना पाता है, ये आइसोऑस्मोटिक यूरिन प्रदान करता है, यानी इसके यूरिन का कॉन्सेंट्रेशन या सॉल्ट का कॉन्सेंट्रेशन इसके एक्सटर्नल रीजन जहाँ पर ये पाया जाता है उस सॉल्ट कॉन्सेंट्रेशन के इक्वल होता है। यह अपना एक्स्ट्रा सॉल्ट अपने नेसल सॉल्ट ग्लैंड के द्वारा रिमूव करते हैं। जैसे हमने बर्ड्स में देखा था कि उनमें भी जो सॉल्ट का बैलेंस है वह नेसल ग्लैंड के द्वारा संभव होता है। तो हम ये कह सकते हैं कि रेप्टाइल, several birds and mammals also do not produce hyperosmotic urine in the presence of plenty of drinking water or where the diet is rich in water. Many of them produces hypoosmotic urine. यानी रेप्टाइल्स में हाइपर ऑस्मोटिक यूरिन नहीं बनता है, यानी इनके यूरिन के अंदर सॉल्ट कॉन्सेंट्रेशन ज़्यादा नहीं होता है। इनमें अतिरिक्त सॉल्ट सोडियम नेसल ग्लैंड के द्वारा शरीर के बाहर निकाला जाता है और इस प्रकार से जो सॉल्ट और वाटर का बैलेंस है वो मेंटेन किया जाता है। ठीक इसी तरीके से ऐसे एनिमल्स जहाँ पर पानी की अधिकता है, पानी उन्हें फ्रूट्स के द्वारा या अदरवाइज़ बहुत ही आसानी से और बहुत ज़्यादा अवेलेबल है, ऐसे प्राणी भी, ऐसे जीव भी हाइपोऑस्मोटिक यूरिन प्रोड्यूस करते हैं, यानी इनका जो यूरिन है उसमें सॉल्ट की कॉन्सेंट्रेशन कम होती है। ऐसे एनिमल्स कई बार जब पानी की उपलब्धता कम हो जाती है तब आइसोऑस्मोटिक यूरिन प्रोड्यूस करते हैं। कुछ बर्ड्स और रेप्टाइल्स अपने यूरिन को क्लोएका यानी एक एलिमेंट्री कैनाल में पक्षियों में पाया जाने वाला एक और रेप्टाइल्स में पाया जाने वाला एक स्पेसिफिक रीजन है, यह अपने यूरिन को क्लोएका में रिलीज़ करते हैं। वहाँ से यूरिन रेक्टम में जाता है और रेक्टम में पानी का रीएब्जॉर्प्शन होता है और इस प्रकार से जो यूरिन प्रोड्यूस होता है वो आइसोऑस्मोटिक होता है। Few birds and almost all mammals have loop of Henle, which is interposed between the ascending and descending part of the nephron that give sight of counter current mechanism. These mechanisms make kidney of the animal to produce hyperosmotic urine when needed. बर्ड्स और कुछ मैमल्स में, जैसे हम मानव में, लूप ऑफ हेनली प्रेजेंट होता है जो कि असेंडिंग और डिसेंडिंग नेफ्रॉन की जो लिम्स है उसके बीच में होता है और यहाँ पर काउंटर करंट मैकेनिज्म के द्वारा सॉल्ट का जो ट्रांसपोर्ट है वो संभव होता है और इस प्रकार से इन एनिमल की किडनी हाइपर ऑस्मोटिक यूरिन बना सकती है जब आवश्यकता पड़ती है। और in above mechanism, the cells in the hairpin tip of counter current loop are exposed to high level of urea and are almost anoxic. They manage by incorporating high level of betaine and sorbitol into their own cytoplasm as counteracting osmotic effect. कई एनिमल्स अपना जो लेवल है ऑस्मोटिक प्रेशर को बरकरार रखने के लिए कुछ प्रकार के सॉल्ट जो है वो अपने साइटोप्लाज्म में घुलित कर देते हैं, जैसे बिटेन, सॉर्बिटॉल। इन सॉल्ट्स या इन केमिकल्स के साइटोप्लाज्म में घुल जाने से जो ऑस्मोटिक प्रेशर है वह एनिमल के सेल का या कोशिका का बरकरार रहता है। Kidney, the excretory organ of vertebrate, it functions both for excretion as well as for osmoregulation. This is a typical structure of kidney. Two pair of kidney present. The right one is bit higher, little bit higher than the left one. The posterior vena cava supplies or takes in the blood from the lower region of the body to the brain. Then there are renal arteries and renal vein. Kidney is a bean-shaped structure. It has got two distinct regions, cortex and medulla. When we take a cross section of kidney, we can see the renal artery, the renal vein interposing inside the kidney, that is entering into the kidney. one carrying the oxygenated blood and the other carrying the deoxygenated blood away from the kidney. Renal artery, oxygenated blood is brought to the kidney by them. Renal medulla is the middle part of the kidney. Renal cortex is the outer region of the kidney. The renal cortex has nephrons, glomerulus embedded in them or present in them. The medulla region has the loop of the or tubules of the nephron. This is a typical structure of a nephron. Nephron organization. Can see the glomerulus, the Bowman's capsule. Inside the glomerulus, can see the arteries and capillaries entering. There is the distal tubule, Bowman's capsule, peripheral capillaries, the arteries and the venules, the ascending limb and the descending limb, the loop of Henle, vasa recta, the collecting duct, distal tubule. It is a typical nephron organization, and as already mentioned, kidney of the vertebrates function both as the excretory organ, that is eliminating the nitrogenous waste in form of urea, as well as they function as the osmoregulatory organ. The nephron is organized for stepwise processing of blood filtrate. The filtrate produced in the Bowman's capsule contains salt, glucose, amino acid, vitamins, nitrogenous waste, and other small molecules. The proximal tubule. The reabsorption of iron, water, and nutrient takes place in the proximal tubule. Molecules are transported actively and passively from the filtrate into the interstitial fluid and then the capillaries. Some toxic materials are actively secreted into the filtrate. As the filtrate passes through the proximal tubule, the material to be excreted becomes concentrated. This is the structure, the proximal tubule, where reabsorption of the filtrate occurs. It is the cortex region where you can see the head of the nephron, the anterior region of the nephron, the glomerulus, the Bowman's capsule, and the proximal tubule where the reabsorption of water and salts occur. Then as the filtrate pass further downward, it is concentrated. Then it passes down to the loop of Henle, where again the absorption of water occurs in the descending filtrate. That is, the filtrate that is going downward, the water from it is again reabsorbed into the body. Then further concentrated filtrate passes, it then ascends into the ascending Henle loop. This region is present in the inner medulla region. As the filtrate ascends the Henle's loop, the salts, sodium and chloride, are again reabsorbed into the body by both active and passive transport mechanism. Again, concentrated urine passes further up Again, the salts, if more salts are required or more water is required from by the body, then the water and salt is again reabsorbed into the body fluid. More concentrated urine, less water and less salt. It passes. This excretory uh, unit contains urea as the nitrogenous waste. If salt is required by the body, the salt is again reabsorbed from the collecting duct, thus making urine more and more concentrated. The water is reabsorbed if water is required by the body. If water is not required by the body and there is plenty of water in the blood or in the body system, the water is uh, eliminated from the system. If water is required, then water is reabsorbed into the system. Likewise, the salt balance is also made. If the salt is present already in excess in the body, the salts are eliminated along with the urine. But if the salts are required by the system, then the salt, like sodium chloride, is reabsorbed into the system. The excretion contains water, urea, and other uh, iron which are present in the body in excess, thus maintaining the osmoregulation or the osmotic balance. The descending limb of the loop of Henle reabsorbs water through the channels formed by aquaporin proteins. The movement is driven by high osmolarity of the interstitial fluid, which is hyperosmotic to the filtrate. Thus, the filtrate becomes increasingly concentrated. Ascending limb of loop of Henle. In ascending limb of loop of Henle, the salt is again reabsorbed. It diffuses from the tubule into the interstitial fluid, that is, into the body fluid. The filtrate becomes then dilute because the salt has been reabsorbed.